So hopefully you can all hear us and see us okay, and we get a good connection today. I see we're still rising in numbers here, up to 34, 35 people. We'll just give it a wee second until this stabilizes, and then we can start. We've another roughly minute to go before we have officially a start time. So folks, just a reasonably good turnout at the moment. See we're up to 65, 66 people here, and we're maybe still climbing. We'll just take a start here, and presentations will be fitted in within the next, I think, hour that we've booked here. We'll hopefully get you back to your normal line of work reasonably quickly. Just to welcome you to start with to the Online Science Seminar Series. This is a seminar series that's been running, it was on and off for a few years. The previous series was run by our colleague Paddy Boylan, and this is a new series we're starting up now. It's the first of a hopefully a monthly series over the next few months. So the topics today are focused on water quality, and I think it's safe to say that Lough Neagh has largely brought water quality to the fore. Lough Neagh is one of the most significant environmental challenges in Northern Ireland, and the recent harmful algal blooms, they're largely tied to water quality issues. And obviously as a department, we have a role in understanding what's happening and trying to look at mitigations, solutions, and also making decisions around what's going to happen. So the science is very much key to this. It's within the Lough Neagh Action Plan. It's stressed very much that the decision making will be heavily reliant on good science. And DAERA has a range of science that it's undertaking around the Lough Neagh issue, and I suppose wider water quality issues as well. Most of you are probably aware of the AFB, the DAERA directed AFB work programme, which is essentially the AFB projects that many of your teams will have maybe policy or science involvement with. But as well as that, we have other areas that we're funding, and they would include DAERA-sponsored studentships, which is Caitlin, who's on the call today, and also the co-centres, and in particular the Climate Plus co-centre, which is represented today by Suman and her work. So our presentations today will cover these elements within the DAERA Science family. So we'll start up with Caitlin's first of all. I'll introduce Caitlin now, and then after Caitlin's talk, we'll have a few minutes for questions, and then we can go into Suman's talk as well. So Caitlin's focus is to develop an emergency procedure for Lough Neagh that destroys toxins produced by algal blooms, as well as the blooms themselves. So with positive consequences for drinking water and the broader natural environment. So Caitlin, if you can take your presentation up, we'll try and stick to roughly 15 minutes and then a few minutes of questions afterwards. And just if people on the call, if you want to put any questions or anything in the chat, we can pick them up at the end of the presentation. So thank you. Caitlin, if you want to just introduce yourself and start your presentation, please.
Perfect. Can you see that presentation okay? Great. So hi everyone, my name is Caitlin and I am a PhD student here at Queen's University Belfast in the School of Chemistry and Chemical Engineering. My primary supervisor is Dr. Andrew Marr, who is on the call today, and as was said before, I am undergoing a DAERA funded studentship for my PhD project, which is titled the Lough Neagh Emergency Development for Novel Treatments for Cyanobacterial Blooms and Toxins. So first of all, as I'm sure you're all aware of the problems at Lough Neagh, the first bloom that was mainly reported happened in 2023, and it has continued to resurface every summer since then. And this is a picture I took during the 2024 bloom that shows how badly impacted the northwest shore of Lough Neagh is during these summers. And you can see that there's a thick layer of the cyanobacteria sitting on the surface of the water. So these harmful algal blooms are caused by an exponential growth in cyanobacteria in Lough Neagh, and this growth has been supported by a few different factors. The first being an increase in temperature during our summers because of climate change, and as well as this there's been an increase in the amount of phosphate and nitrates entering Lough Neagh, which is food to allow the cyanobacteria to grow, and that is why we've been seeing these very clear harmful algal blooms every summer since 2023. This is of great concern to us because Lough Neagh is Northern Ireland's main drinking water source, and without the proper procedures in place to ensure that we can completely remove the cyanobacteria and the toxins that they produce before the water reaches our taps, our access to clean water is at severe risk. So I have a bit of detail about the type of cyanobacteria that cause the harmful algal bloom at Lough Neagh. So in 2023, the bloom was studied by a group here in Queen's in the School of Biological Sciences, and they found that Microcystin aeruginosa, a specific type of cyanobacteria, was responsible for a third of the bacterial DNA reads when the bloom was sequenced. This is just a picture of exactly what this cyanobacteria looks like under a microscope. And these Microcystin aeruginosa cyanobacteria produce toxins within their cells. And so when these toxins are produced, the cells die and the cells lyse out these toxins into the water, which means that they then become waterborne and enter the loch. So this structure here at the top just shows the general structure of what these microcystin toxins look like. And the red box highlights exactly the part of the molecule that inhibits this toxicity. So this ADA group, as it's known, will bind to important proteins in your body, and it disrupts cellular processes, which can lead to irregular cell growth. And this most commonly manifests in the development of liver cancer. So because of the toxicity of the byproducts of these cyanobacteria and also the widespread harmful algal blooms worldwide, the World Health Organization have given a limit of 1 microgram per litre of microcystin toxins to be present in water bodies which provide lifetime drinking water. And this the same group in the School of Biology also looked at the type and amount of microcystin toxins that were found in the 2023 bloom. And in the algal mat samples, so these are just samples of the cyanobacteria that sit on the surface of the water, they were finding at the highest concentration almost 8,000 times the amount of microcystin toxins present. Then also they looked at the surface water samples, so this is areas of the loch which don't have a clear indication that there's cyanobacteria present, and they were still finding at the highest concentration almost 4 times the limit of microcystin toxins present in the water. So this paper really highlighted to us that we needed to do something to safely ensure the removal of the cyanobacteria and toxins from the badly impacted areas of Lough Neagh. And so our research first focused on looking at ways to destroy these toxins and cyanobacteria that are known to us. So chlorine and ozone are the most common chemical treatments, and they're widely used in water treatment and they're well established, but they come with unique disadvantages which make them unsuitable for use in open water bodies. And mainly chlorine has the big disadvantage of interacting with organic material in water to form even more harmful carcinogens, and ozone is very expensive and it usually requires combination with another treatment which requires a very complex mechanical setup. So we look to using hydrogen peroxide, and it's less common, but it does offer a unique advantage for this specific application of destroying cyanobacteria and toxins, and that is that cyanobacterial cells exhibit a sensitivity to really low concentrations of hydrogen peroxide. So we can put very low amounts of peroxide in the water and it will specifically target cyanobacterial cells and leave other cells unharmed. Another big advantage of hydrogen peroxide is it breaks down into water and oxygen, so we don't have to worry about any carcinogenic byproducts like in the case of chlorine. But we did know that there was going to be one main disadvantage that we would have to overcome in order to get the hydrogen peroxide to work as a treatment for the cyanobacteria and toxins. And this main disadvantage is that hydrogen peroxide treatment alone won't work at very large volumes as the concentrations required are way too high. So our research focused on finding a way to make peroxide work for a treatment for harmful algal blooms because we've seen so much potential in its use. And this is when we enlisted the use of an iron tamil catalyst to use alongside the peroxide. So this catalyst was developed by Terence Collins at Carnegie Mellon University in Pennsylvania. And this catalyst's main key features are it supercharges small concentrations of peroxide, which makes the peroxide more effective at combating harmful algal blooms. And in turn, it increases the volume of water that can be treated and drastically reduces the amount of peroxide required to destroy the cyanobacteria and toxins. These catalysts also work at extremely low concentrations, which means that we can use very minute amounts and still activate the peroxide to do the job that we require it to do. As well as this, these tamil catalysts are biomimetic peroxidase mimics, and what this means is that they mimic a peroxidase enzyme that exists in nature and in all of our bodies, and it looks very common to common biological molecules that you probably have heard of, like hemoglobin and vitamin B12, as it has this metal center and it has four ligands attached, and they're all connected by a very strained backbone, and this gives the catalyst the stability that is required for it to do its job. So this is how we were able to come up with the overarching aim for my PhD project, and that is to trial peroxide activating tamil catalysts for the removal of cyanobacteria and cyanotoxins from the badly impacted areas of Lough Neagh. And in doing so, we wanted to make sure that we had no negative effect on the native animal or plant species at Lough Neagh. So a bit of an introduction to these tamil catalysts and how they work. So first of all, we had access to an older generation of these tamil catalysts, and it had been shown to work at destroying micropollutants in water. However, there were some main disadvantages, that being that it had optimum performance at pH 9, which is not the pH that we would require to use it at. So it also had really low catalyst loadings required to do its job, but the main issue with this catalyst was that it was limiting. So once it underwent the reaction with peroxide to destroy the desired molecule, it would undergo inactivation and no longer be active and no longer supercharge the peroxide. So this is how we moved on to using a newer generation, and this new tamil is an improvement on every single negative of the previous catalyst. So the main features of this new tamil catalyst is that it has a kill switch, which ensures that the catalyst doesn't persist in the environment. It has optimum performance at pH 7 to 8, which is great because it's the pH we will probably be enlisting it at. It requires really low catalyst loadings, even lower than the previous catalyst. And at the moment, toxicology tests are currently being performed at the Moat Lab in Florida by our collaborators, and they suggest that tamil decomposition products don't pose a threat to other organisms. So the first year of my PhD, I had one main objective, and that was to obtain some initial results on how efficient our new tamil and peroxide treatment was at destroying the cyanotoxins. So I focused in on the first cyanotoxin that was found in the highest concentration in the 2023 bloom, and that was microcystin RR. So I was able to achieve this objective by going to Robert Gordon University in Aberdeen and visiting Professor Linda Lawton's group, where I was able to analyse the destruction of microcystin RR with our treatment. And the next few slides are going to run through the main results from this trip. So first of all, we did some reactions. The smaller graph in the middle shows our peroxide controls, and I wanted to prove that peroxide alone wasn't powerful enough to destroy microcystin RR, and as you can see, that was achieved. So over almost 5 hours, there was almost no degradation of microcystin RR with peroxide alone. And then in the bigger graph, I had added 100 micromoles of this new Tamil catalyst to the reaction, and I was able to achieve full degradation of microcystin RR beyond detection in under an hour, and with higher amounts of peroxide I was able to get that degradation to be complete within under 10 minutes. So with this, it proved our point that our system does work at degrading microcystin RR, but we want to optimize that system for use, and this would involve lowering the peroxide concentration and also lowering the catalyst concentration. So I was able to lower the catalyst concentration to 0.018 micromoles in comparison to the 100 micromoles used before. And the three lines at the top show the control reactions with peroxide on its own, and you can see that there's no destruction. And with the addition of a very small amount of our new Tamil catalyst, you can see that there's almost complete degradation of microcystin RR in almost under 6 hours. So what this proved to us is that even at very low peroxide and Tamil concentrations, we can still promote the degradation of microcystin RR. So then I tried to lower the catalyst concentration even further, so I went for 0.005 micromoles, which is a very minuscule amount of catalyst added to the reaction. And as you can see again, the control lines show that there's no reaction with peroxide on its own, and with the addition of this really small amount of catalyst, you can see that the microcystin RR degradation begins to happen. And what we found was, as we lowered the catalyst concentration, we then required to extend the time frame that we were monitoring the degradation of microcystin RR over, and we were able to prove what we thought would happen. So in conclusion, the main points of these results show that for hydrogen peroxide, it must remain at a low concentration so that it's specific to the cyanobacteria, and without the assistance of our new Tamil catalyst, the hydrogen peroxide lacks the power to effectively degrade microcystin RR at these low concentrations. And we have proven the ability of our system to catalyse the degradation of microcystin RR. I have a slide about some future work. So the plans for the next 2 years of my PhD is to go back to Robert Gordon University and do some cell studies and figure out how our treatment can work at destroying the cyanobacteria itself. And then after that, we can link it up with the toxin degradation experiments. As well as this, I want to test a few other variants of the microcystin toxins to make sure that our system works well for all and can destroy all microcystin toxins. And then once we perform those experiments, I would like to do the reactions in loch water so that we can optimize our treatment to work in the conditions at Lough Neagh. And then hopefully, if everything goes to plan, we can get some small field trials in line with DERA and AFBI, our collaborators. Thank you very much for listening. I want to thank my supervisors, funders and collaborators.
Thanks very much, Caitlin. I noticed we've actually had a very good turnout for that. I see some people are actually still joining. Just from the point of view of just opening the floor, if there's any questions that you want to ask Caitlin, we can either try and deal with them now, or indeed if you want to maybe email some maybe more detailed requests that we couldn't work with in the next few minutes. Maybe Caitlin can get back to you over due course. Caitlin, just one quick question, I suppose, for myself, and I think Andrew's there on the chat as well, just to kick it off. What do you see as the main market for this technology? Are you looking at sort of water treatment and more control, what I would describe as a more controlled environment, water quality treatments? Is that what you're aiming at the moment?
Yeah, we're exploring both avenues of using it in lock, kind of like in open water bodies in more like badly impacted areas, so a smaller amount of water. But I do see massive potential for this to be used as a water treatment plan, so as a backup to make sure that those toxins are really destroyed before they enter into the drinking water.
And just for myself as well, then, are you aware of any ongoing trials with this in other areas of the world, or indeed you were showing some of the older, I think it's well, the Tamil chemicals. Is it um, has it been widely used, or is it? And I suppose the other thing then is, are there any risks involved with its use?
So we have our main collaborator, the inventor of the catalyst, Terry Collins. He is working in Carnegie Mellon University and he works with a lot of different groups worldwide, but their focus has been on red tide algae and they have showed that the treatment works for the red tide algae, and they also are looking into different types of cyanotoxins. So there's kind of like brevotoxins and marine type toxins that they're looking into as well. So there's a lot going on, but we're waiting for some work to come out on microcystin itself by the team in Florida in the Moat Lab as well. So there's a lot coming out. Sorry, can you repeat what you said just at the end there?
It was just around the potential risks. Are you aware of any risks around potential its use in, if you're looking at it for potentially drinking water supplies or something like that, or if you're aware of any risks around it at the moment?
At the moment, no, but there are toxicology tests being done by our collaborators in Carnegie Mellon. But because we're using such minute amounts of catalysts to do such a big job, we don't, it shouldn't have an effect, but they're looking into it at the moment. So we'll have more information on that over the next year or two when they publish the information.
Thanks, Caitlin. I just wanted some of the questions here I see in the chat. If anybody else wants to pop any questions in the chat, please fire away. Brett Greer is asking what the toxins are degraded to, and has this been studied by LCMS, liquid chromatography?
Yeah, so at the moment we're doing byproduct analysis, so I'm gonna study it with mass spec, and we're hoping to see some similar trends to literature because there have been a few papers published of microcystin degradation with peroxide, but just a different catalyst used, and we're seeing that it will most likely be a very similar mechanism, but we just want to make sure of that. So that work is ongoing at the moment.
That's okay. Um, I see Bernie Core here is asking if you have an intention to look at other microcystin toxins such as LR.
Yeah, so the main research in the literature focuses on microcystin LR, but it's really weird that Loch Ney has a different cyanotoxin found in a really high concentration because most blooms are LR, but for Loch Ney it's RR, and so I thought that it would be good to try and analyse the RR because it doesn't get done very often. But we do intend to look at LR and YR because they were the other type of microcystins found, the top three found in the highest concentrations.
Okay. Paul here's asking just on the commercial availability of the Tamil catalysts. How readily available are they? How expensive potentially as well?
Yeah, so they're available from Sudoc, so they've just set up commercial in Europe, I think. So they're available commercially in America already, but they're setting up for commercial availability in Europe. And I think at the moment they're not to be too expensive, because the whole point of the catalyst is to use them for this purpose of water cleanup. So
Okay. And then I think this is probably the final question here. Siobhan Patrick's just asking how the new Tamil catalysts differ from the older type. Is there anything in particular
yeah,
that sets them
so
apart?
they just changed their structure. So the main thing is that they've changed their structure so that they have different properties because they were finding that their older generation catalysts weren't working well for water treatment because they required a pH 9. So then you had to alter the water before you were able to then treat it and destroy the toxins. So they have focused in on making that work for water treatment processes, so making the pH, the optimum pH for the reaction to 7 to 8. And then they've also focused in on putting a kill switch or a non-kill switch form in. So what that means is that under certain conditions, you can encourage the catalyst to completely decompose. And then that means that you don't have to worry about it persisting in the environment.
Okay. Um, just want to check if Andrew has anything to add, just as your supervisor, if he wants to have a few words just at the end. Then we can move on to the second presentation. If that's
I answered some of the questions, but my answers were very similar to Caitlin's. She did a great job, so I don't have anything to add.
okay. Thank you very much, Caitlin and Andrew, for your time in presenting that. We'll move on now to the second presentation. Caitlin, if you want to remove your screen share, and we can get Suman just in to add hers. Suman, are you on the call at the moment? I don't see a visual for you. You're maybe down the list somewhere.
Yes, I'm here.
That's okay. Just for the people on the call, and thank you very much. See, we're up to 120 here. Suman's focus is to develop an assessment for treating small-scale rural wastewater through wastewater treatment works. It's essentially a nature-based solution looking at short rotation coppice willow. So this is an alternative to direct discharge to rivers, and in theory then should focus on improving the water quality within rivers, especially in the rural environment. Suman, if you want to head into your presentation there, and I think Phil Jordan is potentially on the line as well as supervisor, and potentially can come in for questions at the end. So thank you very much, and if we give you 15 minutes here and see how we get on. Okay, thank you, Suman.
Yes. Can you see my slides?
Yes.
Yes. Thank you very much, Patrick. So hello, everyone. I'm Suman Acharya. I'm a research associate at Ulster University. Today, I will be presenting some result on efficiency of short rotation willow coppice in attenuating phosphorus from small scale wastewater treatment work effluent. This research began in 2019, but I joined the research group very recently. So if you have any queries at the end of this presentation that I'm unable to answer, my supervisor, Professor Phil Jordan, and Chris will be available and happy to answer them. Before we begin the presentation, I would like to thank all the funding agency for their support for this research. Okay, let's start from very basic. What is phosphorus? Phosphorus is an essential element for all life form on Earth, but too much of phosphorus can be harmful for ecosystem. For example, high input of freshwater ecosystem can cause eutrophication, which can lead excessive growth of algae, increased incidences of toxic algal bloom, loss of sensitive species, and can impact on a range of water usage and societal benefit. There are two major sources of phosphorus emission. First one is diffuse source and second one is point source. One of the best example for diffuse source is phosphorus coming from agricultural land, and for point source, phosphorus that comes from wastewater treatment work. In case of Northern Ireland, around 62% of phosphorus in Northern Ireland waterways come from agriculture, whereas 24% come from wastewater and 12% from septic tank. Around 36% of phosphorus coming in waterways, around two-thirds of those phosphorus come from facilities with poor removal efficiency, and that are mainly from secondary treatment and septic tanks, which are common in small and rural communities. So far, many phosphorus removal technologies has been developed. For example, physiochemical treatments, biological treatment, novel technology like membrane bioreactor, river osmosis. but river osmosis, and these systems are successfully used and applied in larger wastewater treatment plant. But translating this system at a smaller scale for a small scale wastewater plant is challenging because of the associated cost, requirement of rigorous monitoring, location of rural small wastewater treatment plant, and variable feed flow from those systems. Despite these challenges, there is necessity to develop simple and effective system with low capital and operating costs to reduce the greater environmental impact caused by discharge from small scale wastewater treatment plant. To address that objective, of finding a small system, the nature based solution can be a best or it can be a promising solution. And among different nature based solution, the use of zero discharge wetland system can be a best example, because this system recycle all the wastewater through evapotranspiration, especially during summer month, and prevent liquid outflow. However, this system has not been extensively studied in case of smaller wastewater treatment plant. Therefore, our research mainly focus on assessing the efficiency of zero discharge wetland system using short rotation willow copies in attenuating phosphorus from small scale wastewater treatment work effluent. This study is running in two sites, one in Kavanaguru, which is in County Armagh, and another in Liscully, which is in County Donegal. These both of these wastewater plants are very small. For example, the wastewater treatment plant at Kavanaguru saw population estimate at 38, and in Liscully it is designed for population of around 60. And the initial stage of this experiment, we collected single weekly sample around midday from upstream and downstream of wastewater discharge point. Due to the inconsistent variation of discharge from wastewater treatment plant, we did not found any significant difference in phosphorus concentration between upstream and downstream side. So we decided to go to the new sampling approach. As a new sampling approach, two automatic samplers were deployed, one again at the upstream and another at the downstream of the discharge point, to collect water sample every hour over the period of 24 hours. The sampling followed 3-day rain-free period every month, especially from autumn to spring. And as soon as the sample is collected, all the samples were brought back to the lab and immediately analyzed for soluble reactive phosphorus using colorimetric method. The early results obtained from autosampler shows that the phosphorus variation at the upstream site over the period of 24 hours is fairly constant, which is represented in the first bar plot here in the up. And in case of downstream site, the early results showed that there was certain rise in phosphorus concentration at certain time of the day. For example, if we see time period from morning to lunchtime around 8 to 1 p.m., there is rise in phosphorus concentration, and again in the evening around 6 to 8 p.m. there was rise in the phosphorus concentration. So this rise in the phosphorus concentration is mm, likely related to increased use of household amenities. And for better representation of these 24-hour data, we crunch them into a box plot which gives um, minimum, maximum, median value along with a good spread of the data. For example, if you see this box plot for the downstream side, this minimum whisker in this box plot, so the minimum value which coincide with the value observed at 3 p.m. and this maximum and this whisker at top represent the maximum value which coincide with the value observed at 7 p.m. Whereas this line at the middle of the box plot represent middle concentration, and the length of this box plot here represent the interquartile range in the data. And the box plot in this slide uh, shows shows data that we observed before the wastewater were diverted before wastewater were diverted to irrigate willow copies. So in this box plot, it gives result for upstream and downstream side, which were obtained from a 24-hour auto sampler. So here you can clearly see this phosphorus concentration is significantly higher at upstream side compared to the downstream side during each sampling occasion. And if we concentrate on the seasonal pattern for both sides, phosphorus concentration it was maximum in July and minimum in March. This seasonal pattern is more concentrated to the flow in our stream, because during summer there was low flow due to less rainfall, and and there is less dilution. Because of that reason, we see that there is highest phosphorus concentration in July and lowest in March. As soon as the wastewater come as soon as the wastewater coming from the treatment plants were diverted to irrigate the willow copies that happened in May 2022, we can see that there is negligible difference in phosphorus concentration between upstream and downstream side. However, there are few disruption. For example, in July 2022 and in August 2022, and again in March 2025 and April 2025. And the reason behind these disruptions are due to pump failure due to blockage. The main question here is, why does pump failure happen? We don't know the reason. It might be due to technical, or it might be non-interest, because these treatment systems are very small. So maintaining this treatment system might be non-interest for the authority. And the box plot here shows result obtained from our second study site, which is Liskuli. So phosphorus concentration in this box plot is very similar to Cabina group pattern wise. For example, if we focus on before irrigation scenario, for each sampling occasion, phosphorus concentration at downstream site is higher compared to the upstream site. And as soon as irrigation began in September 2022, the difference between phosphorus in upstream and downstream site is negligible. It's like more than 95% of phosphorus is attenuated through the willow system. There are few snags as well. For example, in March and May this year, and for this site, we know the reason why there was problem. So there was willow harvesting before April sampling happened. So the use of heavy machinery for willow harvesting obstruct the irrigation system. Because of that reason, there was still higher concentration of phosphorus at downstream side compared to upstream side. But as soon as the problem was fixed, the difference between phosphorus is very negligible. So far, our water sampling is completed, and plans are in place for soil and biomass sampling. For soil sampling, we already have geo-reference location within the irrigated plot. So we'll collect topsoil sample from this georeference location. So those soil samples will then undergo incubation, drying, sieving, followed by analysis of phosphorus sorption index. The observed result will be compared with soil sample which were collected before the irrigation started. So these differences in Sorption index will give us an idea, do the underlying soil receiving wastewater treatment work effluent become chemically saturated? And the second part is biomass. Second part is biomass analysis. For biomass analysis, we will only collect above ground biomass. For above ground biomass, we will select willow plant randomly, but they will be of varying size. Then the willow plant will be chipped. They will be dried, milled, and sieved, followed by phosphorus analysis by acid digestion method. The result obtained from willow biomass will be compared with results of willow that will be collected from unirrigated site in Hillsborough. So this difference will give us an idea, does willow offtake nutrient in their biomass or not. So far, we understand that 24-hour sampling is able to capture periodic nature of wastewater treatment work discharge. Before irrigation, before the wastewater were diverted to irrigate willow stand, the phosphorus concentration was maximum at the downstream side compared to the upstream side. And as soon as the irrigation started, the difference in phosphorus between two sides becomes negligible. That means our system is able to attenuate phosphorus more or less. So our plan of water sampling is finished. So we are planning to collect more soil and willow sampling this winter. And thank you everyone for listening to the presentation.
Thank you very much, Suman. We're just looking maybe at some of the questions in the chat, and maybe start off just with a few of my own, if that's okay. I'm just interested to see, I maybe missed it in the presentation, but have you an idea of what size of willow plot was used for the two different wastewater treatment works? I think you were saying it was less than 500. I certainly on the screen, I think you sort of mentioned there was maybe 60, 70 people in each area. What sort of hectarage or acreage is the willow plot?
Yes, I think for Kavinagru, the population estimate is 38, and for Liskuli, it's 60. But it was designed for 70 people. And in terms of area of wetland, so there are 3 different zones, and each zone is consist of 0.5 hectares. So in total, it's around 1.5 hectare. So yeah, if I'm confused, maybe Phil or Chris will answer that if I have said something wrong.
No, I think that's about right, Suman.
Yeah, okay.
Just checking then too, around the, you've indicated there's obviously a decline or some sort of reduction in P as a result of using the plots. Have you any indication of willow uptake over the course of the year? For example, is there a summer growth period where more P is taken up, or maybe spring growth period where more P is taken up? And then the follow-on question to that is, does the soil buffer P in the meantime? Just interested to know around the sort of the P mechanics within the plot itself.
So for now, once we just have collected water sample. So we do have soil sample result, but before the irrigation started. So our plan is that we will collect soil sample and biomass sample this winter. So once we collect sample in winter and do analyze, then we will have an idea like if there is phosphorus adsorb in the soil or they were uptake by the plant. So at this stage, we don't have an idea like what is happening in the system. And the principle is that the principle of the system is that how the phosphorus will be attenuated. It's because of adsorption in the soil or due to microbial activities, or it will be taken up by the willow. So something should happen there.
No, hopefully I see Jim Motts has a similar question in the chat here. Hopefully that answers Jim as well. We're waiting on further analysis. Just one final question from myself. How long? Maybe you haven't reached the end of the study yet, but have you a rough idea how long the lifespan of the willow plot will be? How long it would remain effective?
Sorry, I don't know the lifetime of the willow.
Can I answer that, Suman?
Yeah.
Patrick. So I mean, the biology of the site and the chemistry of the site, that's kind of unknown at the moment, but there's more practical considerations in that the site is under lease. So before we get to any conclusions about chemical and biological life spans, we have to deal with a lease issue there. I'm just kind of looking at some of the other questions there about Jim's on SRP in winter. Clearly, you know, our concern has been to reduce water framework directive parameter, soluble active phosphorus, during the summer period when flows are low and light is high and temperatures are high. So that's been our major focus. During the winter, what we see is that as the water level goes up, the assimilative capacity of the system, stream system, increases, and we don't see that difference between SRP upstream and down. It's effectively diluted, so it's a summer issue. And we've not really concentrated on any other parameter but SRP because of resource issues at the time, but also because SRP is the one that's in the Water Framework Directive. Our plots irrigated at the same flow rate throughout the year. I think that's right, but what the system is designed as is a variable discharge consent. So it's kind of accepting that during the winter time there's zero evapotranspiration from the site, so water will make its way down from the plot down into the stream system. Now, because that stream system is a higher base flow, then we see that assimilation capacity. So it's only during the summer that it's effectively working as a evapotranspiration site. So we just not too clear at the moment whether the soil is acting as a semi-infinite sink or whether that biomass is taking some of the P off to be taken off the site. So that's part of the, you know, the full life cycle assessment of the site. The question there in terms of other chemicals such as prescription drugs and antibiotics, well, we just didn't have the resources to test that, and clearly that is an implication as well, that the soil and or the biomass is part of that uptake if it's irrigated to those small areas. Comparing with small reed beds, I'm sorry, I'm just going through some of these questions. I kind of know that Suman's not been looking at. She's just been clearly just looking at the chemistry. Yeah, we didn't make that comparison, so I wouldn't have a comment on that. Chris Johnson, I don't think he's on the call, Patrick, but he may have done some work on that. I mean, we just know that the willows, they just really want to grow on these sites. I mean, their growth in a single year could be up to 4 meters. So and then they're harvested and taken off whatever use they're used for. So they just really want to grow. Yes. Was the soil P index known before the willow was planted? Exactly, it was, and that's why we're doing that retest. We did the soil P index, and we also did the soil P saturation index as well. So we have those comparisons to make.
Just checking if there's any other questions.
There we are, Chris.
Oh, Chris is on.
How do they compare with reed beds, Chris?
There's a little. Sorry, I'm going to today, so maybe reception isn't.
You're breaking up a wee bit, Chris, there, but if you can maybe try and continue, we can hopefully get an answer.
Yeah. Sorry. Just a step in, mentioned about other compounds. There's quite a lot of work going on the uptake of the chemical PFOS. So willow and more willow oil systems seem to have a strong effect in actually managing a number of compounds. I think the probably positive answer to that. The only that I was going to fill or add the conversation was Simon referred to a couple of breakdowns and issues with the system when it wasn't operating, and that is a problem. Unfortunately, the willow irrigation systems that have been constructed in Northern Ireland and Ireland over the last 10, 15 years have all been constructed under European funding, grant funding, project funding. And when those projects there, it's difficult to maintain, difficult for them to be maintained and organized. And therefore, unfortunately, breakdowns. So that really that if anybody was asking that question, that is something which is which needs to be dealt with, the light leasing aspect, which Phil mentioned soon.
Chris, you broke up there quite badly, and I think you're sort of indicating a need for sort of future funding around Willow establishment and maintenance of site. So thank you very much for that. Just want to check if there's anything else maybe from Suman, Phil, and possibly Chris, if we get a good line there, that you want to mention around this. I'm assuming that there will be some linkages at the moment to long-term practical uses of this, and maybe some sort of investigations with maybe NI Water or similar private firms who may have wastewater issues. Is there anything you want to say along those lines, Phil?
Well, I guess, what we're seeing from the irrigation from, just being observing this as a passive scientist, the data look pretty good. I can't think of a nature-based solution that I've come across that has up to 95% phosphorus attenuation. It's pretty good. Now, there's a solid question about how long will that last, and hopefully some of the data we'll be doing in the winter will show us that. I mean, the change in phosphorus, the saturation is the kind of key thing. Chemical saturation is the key thing, and if we get in any export of PE in the biomass, that will buffer that soil P saturation. The biggest issues I've seen so far are to do with I suppose poor maintenance, and it's not to do with the willow, it's to do with the bits between the willow and the house. So it could be mechanical, but maintenance is the big thing, and that's at both sites. That's between the site that's operated by Donegal County Council and also the site that's operated by NI Water in County Armagh. And I think because they're small, I think we know from the very start there is a clear there's poor importance placed on small systems, despite there being many hundreds in the environment, because they're not large urban systems. But when we scale those small systems up, they're a significant source. And up to now, and we haven't got any kind of nasty photographs or scratch and sniff images, but they are literally direct discharge of poorly filtered and so partially secondary treated effluent. It's the outfalls are covered in surfactants, and it's literally kind of semi-raw sewage that's going into the system. So anything that treats that in any way has got to be positive.
Thank you. Just see a few more questions in there. One from Gareth Greer, just around the estimated land area. Assuming, I think you said it was 1.5 hectares for around 60 to 70 people. Hopefully that's covered it. This in terms of the treatment system, were the systems the same, assuming, just in terms of filtration, and essentially are you getting the same inputs into both sites?
Identical, yeah, they're identical.
Okay, right. And then one from Hillary, is this transferable to reduce P from agricultural runoff?
Chris will be able to answer that one, Patrick.
It's okay. Chris, I'm not sure if you can still hear us or not, but I'm aware of work that you've done previously around use of willows in agricultural P reduction, general nutrient reduction.
I think on the Hillsborough sites are showing this where the plots have been put onto critical source areas. Known critical source areas have been mapped with the new system that we have. So I have not seen the data from those yet. Chris will have those data.
Yeah, that's fair enough.
But in principle, yes. In principle, absolutely, yeah.
Okay. If there's no other questions,
and they have also tested the flow coming from dairy farm. They have tested this system for the dairy farm as well, right?
Yeah, I think so.
Yeah.
Yeah.
If there's no other questions, Suman, do you want a last word on this, just around maybe where you hope to take the project in the next weeks and months?
Sorry, can you repeat the question?
It was just where you would hope to go with the project in the next few weeks and months. What are your sort of your future hopes for this work?
Yeah, yeah. Just to follow that up, Suman. So Suman will be completing the soil and the biomass work during the winter, and we'll finish this phase of the work in around about May time. So there'll be a report and hopefully a publication beyond that time. And then we just have to stand back, Patrick. It's NI Water's responsibility to keep the system running, and we can just make the recommendations.
That's grand. Anything to add to that, Suman, at all?
Yes, and it would be interesting to go back to the system after a certain period of operation, so that we'll have an idea how well the system work for a small wastewater treatment plant. Because if we collect the soil sample and biomass sample again after like 3-4 years, then they will give us an answer whether the soil is become chemically saturated. Are they still able to uptake in soil or biomass as well? So it would be interesting to go back to the site after certain year.
No, that's beneficial. I'm sorry, I think a final question here from Gareth Greer. Just would the uptake of this type of treatment be increased by adding nutrient parameters to consents for small works?
Yeah, should maybe try that one, Gareth. I don't know.
We don't know.
That's fair enough.
Yeah, with that one, Phil, yes.
No, if that's I think a very good presentation. I think if that's all from our audience and yourselves, maybe try to wrap it up for today. Thank you to Phil and Chris, who are the supervisors. Suman, thank you very much for a good presentation. Very interesting, and it's nice to see practical implementation of some of the science and research that's going on. So thank you very much.
Thank you.
Okay. Just to say again, this is the first of a series of monthly seminars, and we'll hopefully be back after Christmas with another topic. Each topic should hopefully have its own presenter and set of circumstances around which it's presenting on. We'll get back to you in the next few weeks around that, and we'll try and keep this organised over the next few months. So thank you very much for your participation today, and hopefully you benefited from it. Much appreciated.
Thank you.